Hello, I'm Joanne Geer. I'm the Executive Director of the Biopharma Research Council. We are an association for scientists from academia, industry, nonprofit, government, and supplier laboratories. Just want to note that today's session is being recorded, and you'll see on the right-hand side a question box where you can add your questions for the speakers at any time. Just going to tell you a little bit about some upcoming events that we have. So we're in the series, the short course in the exosome, which I'll say a little bit about in a moment. On October 28th, we're going to be in North Carolina in Research Triangle Park for the Triangle Biotech Research Symposium, a really interesting one-day program. We have a webinar in November, on November 20th, on the emerging FDA guidance for devices intended for home use. This is part of a series that we do called The Mobile Patient. And in March 25th to the 27th, in a really lovely retreat center near Princeton, New Jersey, we're going to be doing the 6th version of Best Practices in Outsourcing with a focus on preclinical and clinical. Everything, of course, is on our website. Just going to repeat for anyone who's new that you can always add questions through the question box on the right-hand side of your dashboard. Just want to thank everyone. We have a number of partners that are making this program available, and it's been a really terrific collaboration. Just Winston Kuo, who we'll meet in a moment, really put together the whole series. I want to really appreciate his involvement and contribution. Last week we had Jan Lutval, and the recording of that webinar is already on our website. Today we're going to hear from Johan Skog. Next week will be Sasha Vlasov and Angel Ayuso Sacido. I hope I'm saying that reasonably well. And one or two other speakers will be discussing the regulatory environment and funding on October 2nd. Winston, I'm going to tell you just a little bit about Winston, and then he'll take it from here. Winston Kuo is the COO of IES Diagnostics, developing diagnostics based on a technology licensed from the FDA, focusing on systemic lupus erythematosus. He's also editor-in-chief and founder of the Journal of Circulating Biomarkers. In addition, he sits on the scientific advisory board of Exosomics and Hansa Biomed, both focused on exosome-based diagnostics and life sciences products, respectively. His research interest in extracellular vesicles include the technical aspect of commercializing both from life science and clinical aspects. Winston.
Thank you, Joanne. Hello, everyone. As I said, my name is Winston. Not only the Chief Operating Officer of IES Diagnostics, but also the Editor-in-Chief of the Journal of Circulating Biomarkers. And I'm very delighted, in collaboration with Biopharma Research Council and Intech Publishers, to provide a short course of an exciting area of extracellular vesicles, the transition from tissue to liquid biopsies. As Joanne had mentioned, we had our first session with Dr. Jan Lutval last week, who gave an outstanding and informative presentation on the overview of the field with the latest, including unpublished data. And you can access his presentation and talk using the link on the Biopharma Research Council site under the short course in extracellular vesicles. Dr. Johan Skog we have today to discuss in his session the up-to-date applications of extracellular vesicles. And why is this important? I mean, extracellular vesicles, including exosomes and microvesicles, has shown to carry a variety of macromolecules, including mRNA, microRNA, other non-coding RNAs, lipids, and proteins. And within the past 5 years, they really have emerged as a promising, minimally invasive, novel source of material for molecular diagnostics. So although extracellular vesicles can be easily identified and collected in a variety of biofluids, a lot of research and proper validations is still needed in order to bring them, to fruition in a clinical setting. So there are 3 live sessions remaining, including today. The one on the commercial implications presided by Sasha Vlasov next week, and the one on the regulatory aspect. So today we're really excited to have Dr. Johan Skog a pioneer and leader in the field with vision, who is currently role as CSO of Exosome Diagnostics, translating the clinical applications and utilities of extracellular vesicles into the clinic.
Winston, we've lost you.
Can you hear me? Hello?
We do now, yes.
Okay, sorry. So I was just mentioning that we're excited to have Dr. Johan Skog, a pioneer and leader in the field with vision, who is currently CSO role at Exosome Diagnostics, translating the clinical applications and utility of extracellular vesicles. So during his time at Mass General, Harvard Medical School in Boston, Dr. Skog discovered that tumor-derived mutations can be detected in microvesicle RNA from serum and other biofluids, which was actually published in Nature's Cell Biology in 2008. Additionally, he demonstrated that microvesicles, serve to deliver messages to other cells, including favorable to the proliferation of cancer. His research background includes virology, gene therapy, brain tumors, RNA, and biomarker discovery. He's an inventor on several patents. So a few housekeeping items before we begin. Dr. Skog's presentation will run about 30 to 35 minutes, after which we will open up to the audience for a question and answer session. You may, however, submit your questions during the presentation by entering the questions on the dashboard to the right of your screen. So thank you very much, and now I'll hand it over to our speaker, Dr. Johan Skog. Please enjoy.
Hello. So just as we do this, we do have the opportunity here to make to do a very quick poll, which I'm going to launch right now, just a few questions, and then we'll begin our presentation.
Okay, thank you so much, Winston, for that introduction, and welcome to the second session of this webinar series. You have to bear with me a little bit today. I'm suffering from a bad cold, but I hope you guys can hear me all right anyway. So I'm going to talk a little bit about the exosomes, how they can be used, and why they are important for diagnostics. But I will start by talking about the sample collection and the processing that is important to get to the result that you need for the clinical diagnostic. I will end by speaking about the platform applications involving exosomal RNA mutation analysis, as well as exosomal RNA expression profiling. So the genesis of exosomes or microvesicles or EVs were extensively covered on last week's session, but for you who did not attend, I will give you just a quick recap of that. Microvesicles are released by many different mechanisms. One is the classical mechanism is through the endosomal pathway, where you have formation of an endosome and then inward budding into the endosome, forming these multivesicular bodies.
Johan, this is John.
Yes.
Could you just share your screen? I don't think we're seeing the screen right now.
Oh.
I think the poll interrupted it, so you might have to reshare it.
Okay, can you see it now?
Definitely. Thank you.
Okay.
All right. Okay. So moving along here, let's see. Now my screen froze, of course.
Just make sure you have the PowerPoint window selected and hit that.
All right, hang on here. Let's see if this works now. Yeah, I can not move it when I'm sharing the screen, actually. It works well when I pause the screen.
Let's try unsharing and resharing.
Mm-hmm.
Looks like you're still sharing. It hasn't disappeared. Let me
It says it's paused over here on this
Right. So
side.
you want to, 1 second. Let's try it again.
Okay, can you see my screen now?
Yes.
Okay. Unfortunately, it freezes my screen when I go into presenter mode. I don't know why this is. This is unfortunate.
Can we send
Johan,
a
this is
PDF?
Joanne. Would you like to, why don't you forward them to me and I'll run the slides for you?
Sure.
That's easy to do. Or to Winston, but I'm happy to do it.
Sorry about that technical failure here.
It's quite all right.
And Joanne, just let me know when you have it and I'll share your screen.
Will do.
Okay, I can continue speaking in regards to the screen that you're seeing now, I guess. Okay. So microvesicles are released by different pathways. The most classical one is the multivesicular body pathway that you see here to the left, releasing the vesicles to the surrounding. You also have direct budding at the plasma membrane, releasing vesicles. And currently, there's no consensus in the field on surface markers that can identify vesicles of different sources. So they have been referred to as exosomes or microvesicles, microparticles, but they are really extracellular vesicles, and we don't know how to discriminate the different types of the vesicles currently. So in this presentation, I will refer to all extracellular vesicles as exosomes.
Unfortunately, they haven't come through yet. They didn't go to spam.
I suggest that we discontinue and let Johan just go ahead and we'll put it up later and make it available to the audience.
I am seeing your screen. Large vesicles released from primary tumor stem cells.
Yes. So can we go ahead in this presentation mode instead of the?
Yeah, that's fine.
Okay.
Yeah, let's do that.
Okay. Okay. Sorry about that. So we have many different types of vesicles being released from cell types. But what's interesting is that stem cells seem to have a lot of these larger vesicles. These are pictures of primary glioblastoma stem cells expressing CD133. And you can actually see that a lot of these larger vesicles are budding off from these cells. So extracellular vesicles involves a very wide range of vesicle sizes, anything from 30 nanometers in diameter and up to actually several micron. Although in biofluids, I would say that the absolute majority of the vesicles range around in the 100 nanometer range. When you characterize exosomes, you can study them by EM. At the top here, you see a primary GBM cell analyzed by scanning electron microscopy. And you can see a lot of these microvesicles being released from the plasma membrane. And if you isolate these exosomes, you can embed them in plastic EPON and cut through them and do transmission EM microscopy. And when you do that, you can see that vesicles in general are a very heterogeneous mix and they vary a lot in size. You have the very small vesicles around 30 to 50 nanometers, as well as larger vesicles just over 100 nanometer in diameter. Okay, so the sharing seemed to be back on track here, so I can show you in presentation mode. When you do the imaging by cryo-EM, you can see that the morphology of the membrane of this these vesicles are it's a double lipid membrane, but interestingly is that you have, well, the majority of the vesicles are single lipid bilayer membranes, but you do also have multilamellar vesicles with vesicles within vesicles, as you see here to the right. What's interesting with these exosomes is the actual content. They contain a lot of different things. They contain mRNA, microRNA, and other non-coding RNA, as well as interesting proteins like the MMPs, the angiogenic proteins, tumor-specific proteins, and in the case for neurodegenerative diseases, they carry tau proteins, alpha-synuclein, and even prion proteins. So they are a really good platform for biomarkers. In this talk, I will mainly cover the RNA part of the exosomes. What's important to know is that these vesicles, they carry the entire or most of the transcriptome, but not in every vesicle. The vesicle is very small. I said that the majority is around 100 nanometer, which is similar to a retrovirus in size. And we know that the packaging capacity of a retrovirus is only around 10 kb. So logically, you cannot package the entire transcriptome into one vesicle. It's more likely that you have one or a few RNA transcripts per vesicle. And since when you isolate the vesicles, you typically look at the result averages of billions of vesicles that you have in your prep. So each individual vesicle is different and carry a different message. So what is the advantage of using the exosomes to get to the RNA in biofluids? When you do the RNA extractions from biofluids, you can either isolate the exosomes or other subcomponents of the biofluid first and then do the RNA extraction. Or you can go ahead and do what we call direct RNA extraction. And here I'm going to show you an example where we do direct RNA extraction using a commercial kit for RNA extraction from plasma. And we are out of presentation mode here. Unfortunately, it's no longer working here.
You try clicking on the slide, it doesn't do anything?
There you go. So when you do the plasma, can you see my screen? Or is
Yes,
it paused?
I'm not sure. I think you paused it.
Can you see it now?
Now I can, yes.
Okay. So when you have a serum or plasma sample or other biofluid, there's a lot of different components inside. You got a lot of nucleic acid, free-floating nucleic acid, mostly DNA. You also have vesicles carrying RNA inside. There are recent descriptions of cholesterol carrying RNA in biofluids, like HDL particles. And you also have Argo2 or the Argonaut 2 proteins carrying microRNA in biofluids. So when you do extraction using the exosome platform, it enables you to subfraction the RNA from the different cellular processes You can do a total exosome extraction using either the classical method of ultracentrifugation or filter purifications. You can also use affinity purification to capture specific subtypes of vesicles using magnetic beads or microfluidics or other means. With a whole plasma extraction or direct extraction, the problem is that you don't have any selectivity of the subfractions in plasma, and you get RNA from all kinds of components in the plasma. And there's also a limit in the efficiency of the RNA extraction when you go into higher volumes, because when you do direct extraction from larger volumes of plasma, it does not seem to work very well for RNA due to all of the RNase activity that can be found in biofluids. This is a bioanalyzer example where we compared direct extraction of a 2 milliliter plasma sample using either an exosomal isolation method, in this case it's the exoRNEC extraction method, versus a direct extraction shown in red. And you can see that the intensity of the peaks are very, very low from the direct extraction due to the fact that when you have larger volumes of plasma, that's like 2 ml, the residual RNase activity in the plasma is not properly inactivated. So here you can see with the exosomal platform, you can purify the vesicles from the RNase activity, and you'll have a nicer RNA preparation with intact 18 and 28S ribosomal RNA, as well as a very large low molecular weight RNA peak shown here to the left. This low molecular weight RNA peak is not only microRNAs, it's a wide variety of different structural RNAs like tRNAs and other non-coding RNAs. When you do your extraction, it's actually very important that you can have a high reproducibility from the same sample, and it's also important that you can scale it, because I'll cover that a little bit later, but it's very important that you're able to extract from the volume of plasma that you need for your purpose. Here you can see an example with the reproducibility of 2 extractions overlaid on top of each other from 1 milliliter of serum. And below here you see the same thing, but from 3 milliliters of serum. And when you do the analysis, the downstream analysis of this, you can see that you detect more RNA species if you extract from a larger volume, and you also get lower CT values in this range, indicating you'll have a more robust measurement of your sample. So going over to the sample collection and the pre-processing of your sample, this is a very important point when you do biofluid extractions. So pre-processing of the sample is something that you need to do to ensure that you don't have, for example, cell carryover into your preparation. So a typical flow of an exosome isolation method is when you take your serum sample or your plasma sample and you do either a low G spin or a filter approach. Sometimes that pre-processing is repeated by increasing the G force, and then you take the supernatant for your exosome isolation. But it's very important that you have a standardized protocol for the collection and the pre-processing, and you have to understand what happens in each of these steps. So the first one is the collection tube and the additives that you have in it. So it depends if you want to have a serum sample or plasma sample, and I'll show you some data later that it really matters how you process your sample. The processing time is also very important. The handling of the tube. For example, if you draw a blood tube into a plasma tube, it's very easy to get activation of thrombocytes if you drop the tube or you treat it in the wrong way or leave it on the bench for too long prior to centrifugation and processing of the sample. You can also have cell carryover into your plasma if you're not careful with the method. And that's why we use this pre-clearing step. But it's really a balance between the vesicle loss and the carryover of cells thrombocytes in the pre-clearing step. And thrombocytes also carry RNA, so it's important to understand the contribution in your sample here. Most people that work with exosomes in plasma, they use platelet-poor plasma. But even platelet-poor plasma can carry up to 10,000 or even more thrombocytes per microliter of plasma. So it's a very abundant entity in plasma. It's also known that many RNA-containing vesicles pellets at lower speeds than the traditional exosome pelleting conditions. So when you do your low G spin or your filter, you have to weigh. It's a very it's a balanced approach here to either have a little bit more vesicle loss and less carryover of other compartments into your sample. Versus losing yield. We also know that many of the membrane vesicles have overlapping diameters to platelets, which are typically around 2 micrometer in diameter. I showed you that on an earlier slide from the tumor stem cells, that they have a lot of larger vesicles. So this is a very important consideration when you do your isolation. How much you are willing to lose of your titer in your pre-processing or pre-clearing step. And when you do standardize your protocol, do not only report centrifugation time in G, because pelleting efficiencies of different rotors are very different depending on their K factor and will give you very different results. So when you look in the literature and you're trying to replicate a protocol, and only the centrifugation time in G is noted, you have to make sure that you're using the same rotor as well, because this is very important. The next step is the actual nucleic acid biomarker characterization. And to characterize the nucleic acid that you get in your sample, you have to understand if you have DNA versus RNA, or what's the contribution of both of them in your sample. So if you want to do nucleic acid yield determination for transcription profiling, you need to include a control for DNA. Because these extractions from biofluids typically yield fairly low amounts of RNA, and they're easily biased by DNA. And there's a lot of cell-free DNA and other DNA components that can co-purify in your preparation. So that's an important consideration in your analysis. You also have to choose an extraction method that fits well with your needs. You have to understand, you have to know what is the marker that you're trying to detect and what is your normalizer. And this is something people often forget about. The normalizer is as important as the marker. And you have to understand what the copy number of this biomarker and normalizer is per milliliter of biofluid. When you have figured that one out, then you have a benchmark for how many milliliters or what the volume of the biofluid is that you need to do a robust detection. So you can calculate the appropriate biofluid volume needed for the detection, and then you need an extraction method that allows you to scale the volume of the biofluid to the appropriate volume. You also have to understand your marker that you're looking at. Is the marker present in other compartments in the biofluid? Is the RNA that you're looking at present only in exosomes, or is it carried also in the AGO2 fraction, in the HDL or platelets, etc.? Because more RNA is not always better. These compartments in the blood are coming from different processes, and if you're studying a certain process and a certain marker, you don't want that to be biased by the addition of the marker from another process in the blood. And when you understand your marker better, then you can build that into your pre-processing strategy for the sample. So the exosome isolation method that we currently use here, and that I will show you in the next set of slides, is the exoRNA extraction. So the reason we chose to use this method is that it's scalable between the volumes of 100 microliters up to 4 milliliters, and the procedure is fairly quick and use disposable spin columns. The flow of the isolation is that you add the vesicles to the membrane, you bind them to the membrane, and you wash. And then you do actually a lysis on the filter to release the RNA. And after that, it's a fairly standard RNA extraction protocol that gives you a nice clean RNA sample at the end. So this analysis was done using the exoRNA kit, and this is actually to study the effects of different blood collection tubes. And most blood collection tubes are compatible with the extraction, but they generate different biases. For example, in a serum tube, you can have an addition of thrombocyte markers due to the activation of the platelets in the coagulation process. This is a very busy slide where you see the PCR CT values of all of these genes that you see to the right. At the top you have mRNA genes, and at the bottom you have microRNA genes. And as you see here, they follow similar trends, but they are different. So one CT value is a twofold difference in starting material. So you do have fairly significant variations in your result depending on what tube you're using. And that emphasizes the need to standardize this and have a very well-controlled collection protocol for the study that you're using. And you have to ensure that all patients have been collected under the same protocol. I don't want to give a recommendation for a particular tube type because they all work and they all have different biases. One thing I have to mention a caution about is the heparin tubes, because heparin is a known PCR inhibitor, and as you can see here, there are absolutely no results coming from this tube, and this is not due to lack of RNA. The RNA is still there, but the PCR does not work when you have heparin in the tube. The exosomal RNA is really stable once it has entered the plasma, and the plasma have been pre-processed into the right format. Here at the top, you can see the stability of the exosomal RNA after incubation of plasma, 25 degrees over time. And you can see that the CT values are very stable. And the same is true for freeze-thaw cycles, where you have 9 consecutive freeze-thaw cycles here, showing a very stable result of the data. That being said, we still recommend you to have a fixed number of freeze-thaws, preferably aliquots, and only freeze-thaw them once to have a baseline in your analysis. Just because you never know what happens with certain patient samples when you do your study. I mentioned previously that there are multiple sources of RNA in biofluids, and it's important to understand that the exosomal RNA is not the same as direct precipitation of RNA. This is an example where we compare the microvesicle extraction with direct extraction. And this comparison can only be done on small volumes because direct extraction has an upper limit around 200 microliters of plasma. So we can't really do this at larger plasma volumes. But you can see here that the ratios between different genes are different. So if your study is looking for a ratio of two genes, you have to ensure that the isolation method is the same, that you use either direct precipitation or the exosomal fraction, because they come from different sources and they contribute to your result. This is showing you that with the exoRNA filter, you can actually capture what's inside the vesicle by doing the lysis on the filter. And you can see that you have all of these mRNAs here, as well as the microRNAs. You can also capture the flow-through and isolate the RNA from the flow-through. There don't seem to be any mRNA remaining in the flow-through, so all the mRNAs are captured on the filter. It's only a very low copy number here up at the top. But there are certain microRNAs that are very abundant in the flow-through. Some of those microRNAs have previously been characterized to be bound to AGO2 proteins, for example, miR16. You have other microRNAs that are known to be preferably in the vesicle fraction, like Let7. And Let7 is in orange, or sorry, it's behind here, I think. So you can see that the capture of the vesicle fraction is very efficient. Many of the microRNA markers exist in different ratios in both components. So high volumes are needed for high sensitivity applications. Many of you are probably starting with screening studies. So here we show you the result looking at oncogenes with microarrays, so looking at 90 different mRNAs involved in cancer. And when you do the extraction from 200 microliters of plasma, the majority of the mRNA genes are not even detected. They're above the threshold. So here you see 46% of the genes are in the CT value range that is not very accurate. If you scale the volume and you go up to 1 ml, 2 ml, or even 4 ml, you get significantly lower CT values, which gives you better detection or more accurate result reporting, as well as more genes detected. You also need high volumes when you do tumor mutations. When you're looking for tumor mutations in blood, it's typically a rare mutation. It's the majority of the vesicles in circulation or in plasma is really from normal processes, and only a fraction of them are from tumor exosomes. So here we actually did deep sequencing of KRAS from a plasma sample of a colorectal cancer patients. So we took 2 ml of plasma, and in this particular patient, 10% of all the KRAS reads that we got were mutated. So that corresponds to the fraction of tumor exosomes or tumor KRAS in the sample. In many other patient samples, it's even lower than that. So it's really a rare event. So, and if you're not able to scale your volume to the appropriate volume, you will not detect many of these mutations. So one question we often get is the exosomal RNA profiling able to monitor treatment response? So here we looked at mRNA as well as microRNA profiles in longitudinal serum samples. First, we wanted to understand the reproducibility of the sample extraction and the actual RT as well as the qPCR. And we assayed here about 750 different microRNAs, and we were fairly happy with the reproducibility of the actual process. So we went on to analyze serum samples from brain cancer patients that were enrolled in the drug clinical trial. So we had 100 frozen archived serum samples, pre and post treatment, and we extracted them. In this particular case, we had about 1 milliliter of frozen serum. And in that sample, we detected roughly 500 or so of all the mRNAs that are involved in this oncology panel that we screened for, and about 300 or so microRNAs in each sample. What was interesting in the screening approach here was that we saw some unique expression changes in the responder groups. And in particular, here you can see the result from microRNA 10B that seemed to go down in the responder group, but not in the non-responder group, which is very interesting because microRNA 10B is a known microRNA to be upregulated in glioma patients. So we actually went on and we did a singleplex validation of the miR10B in the non-responders versus the responders, and we saw that the responders seemed to all have a drop in the miR10B level after treatment, but not the non-responders. And if you plot the data to the progression-free survival, it seemed to correlate fairly well. So why would you measure a tumor mutation in biofluids? It's known that tumors are very heterogeneous, and treatment decision and outcomes are linked to the molecular characteristics of the tumor. And they are also very dynamic and change over time. One typical example here is in non-small cell lung cancer, where activating mutations in EGFR can give you a response to treatment with EGFR inhibitors, but soon after that treatment, you often develop resistance mutations like the T790M. So it would be beneficial to be able to monitor this longitudinally over time and see how a mutation changes. It's also less risky for patients than an invasive biopsy and cheaper, and it's more practical to do the longitudinal samples of biofluids rather than in biopsies. However, the challenge here is to isolate and detect these rare tumor transcripts in the background of normal. So here it requires you to do something with a high sensitivity. So we've chosen to go for targeted sequencing, and we have a panel covering most of the, or many of the interesting genes that are mutated in cancer. And when you do targeted sequencing, you do a very deep coverage of each of the genes. And when you do that, you can start to see these mutations popping up. So here, for example, is the second most common read. The most common read is the wild type of BRAF, for example. So we had a malignant melanoma patient, and we extracted the exosomal RNA from the blood, and 1.8% of all the BRAF transcripts that we sequenced carried this V600E mutation. In other patients, the level can go up as high as 10% or even higher, but that's rare. Most of the times, the fraction of BRAF is lower than that. A 10% BRAF mutation ratio would correspond to 20% of the BRAF transcripts coming from the tumor, because in this case, it's a heterozygote mutation, and we know that the mutations are being packaged in a 1-to-1 ratio, the wild type and the mutant, into the exosomes. And in this particular case, we did the sequencing of EGFR exon 19 in a non-small cell lung cancer patient, and we can see this deletion here in this region corresponding to the exon 19 deletion. And we also plot, so at the top here you have the sequence of the most common read, which is the wild type. The second one here is the deletion. And then you have the third and the fourth and the fifth, etc. Most common read here, and they all have small variations in the sequence. And with that, I want to thank you for your attention and apologize for the technical difficulties we had in the beginning. And I also want to acknowledge all the people that have been involved in the different projects here.
Thank you, Johan, for such an outstanding and informative presentation. I just want to let everyone know that are still online. We're still continuing to accept questions, and please continue to submit them on the dashboard. And if we do not have time because of the technical difficulties earlier, we will also ask Johan perhaps or our team to be able to provide answers to those, and we'll provide a link for the webinar and along with the written responses to the questions. So please continue to submit your questions. So let me begin. We have a question, several questions. One of which is, what are the factors influencing the harvest of RNA, and what concentrations of RNA are typically obtained from plasma?
That's a great question, and there's a lot of things that influence the amount of RNA that you get. One is, of course, the disease and the actual plasma sample that you're collecting from. Another is the way you collect it. For example, more is not always better. You can take a blood tube and you shake it before you process it, and that will activate the platelets and other things, and that will give you a ton of RNA. But it's all irrelevant RNA because it's processes that happen post blood draw. Typically, you get somewhere around 20 or so nanograms of RNA from a milliliter, but it varies a lot. It's not there's a high variability in the amount of RNA you get, and it's also depending on the way you measure the RNA. If you're using a bioanalyzer or another measure of your RNA, we typically look at copy numbers of certain RNA transcripts rather than fluorometric or fluorometric methods to quantify the RNA.
Okay, from the same person, I want to keep it the same, and they asked very good questions last week as well. What is the minimal amount of RNA needed to do deep sequencing?
Right, so that depends on what you want to sequence. RNA is not like DNA. So in DNA, every gene is a 1-to-1 ratio. So if you take 10 nanograms of DNA, you have the same copy numbers of everything, and you can have an even coverage of it. For RNA, that is not true. You can have 5 or 6 or even more logs, different levels of different RNAs. So if you take 1 nanogram of RNA and you do deep sequencing, you can get good results, but you will not see the rare transcripts. The more RNA you use as input, the higher diversity of RNA you will detect. And most of the times you would like to look at RNA in the low abundance region, because the most abundant RNAs are maybe not relevant for your applications because they are typically ribosomal RNA or tRNA or other structural RNA.
So I want to say that the two questions, Johannes, from Alan Ezrin, and he says thank you. So the next question, could exosomes be a vehicle for metastasis and therefore be markers for metastasis as well?
They might. There are some papers indicating that, but I think it's very early days to say anything about that. We'll have to follow the field and see how it develops.
Okay, another question. Are there any suggestions on how to get rid of the fat that floats to the top of the ultracentrifugation tubes when isolating exosomes?
Right. That's one component that should be built into your standardization, because if you have fasting blood versus a patient that just had a meal, the plasma will look very, very different. In most cases, when you have a fasting blood sample, you don't get this cap of fat, so that will eliminate that problem. Otherwise, I would just recommend to avoid that lipid cap on the to not put that into your sample.
Thanks, Johan. Another question. Is it possible to analyze not just the presence or absence of specific mutations, but the expression of a given marker? Are there reliable, normalized markers in exosomes to evaluate differences in expression?
Yes, I showed you a few slides here where we could monitor expression levels as a response to therapy, and we've also done analysis that we published looking at RNA expression levels in glioma patients. The normalizer is very important here, and there's no universal normalizer that we can use. Like I said in the beginning, an exosome is not like a cell. In a cell, you have all of the transcripts coexisting, and you have housekeeping genes that you normalize to. Whereas in exosomes, they are so small, they package very little in each vesicle. So you're looking at the average transcripts of billions of vesicles. And when you do that, it really depends on the disease that you're studying. So you'll have to find the normalizer that works for you. There's no standard that you can take. You'll have to make sure that the normalizer is doing the job in that population that you're studying. We have different normalizers for different diseases. When we extract exosomes from urine, for example, we have one normalizer for the prostate cancer assay, and when we do extractions from blood to monitor treatment response, we use a completely different normalizer.
Okay, a question related to the kit. In addition to the serum and plasma, can the ExoRNA-EZ Maxi kit be used for other sample matrices like fecal samples?
That it's not supported in the current format, but if you stay tuned, there might be developments there over at QIAGEN. I can't speak for the development efforts there.
Okay. Another question: can exosomes be isolated from blood collected in cytochecks tubes? And do you know the quality, the RNA quality afterwards?
Yeah, I'm sorry. Say what tubes?
Can exosomes be isolated from blood collected in cytochecks tubes?
I don't know. I haven't tested that.
Okay.
Most tubes are compatible, but they all have their own biases, especially if you have activation of certain cell types in there. But as long as all of your samples are collected and processed in the exact same way, and you infer the same type of bias on everyone, it might work for your purpose.
Okay. A couple more questions. Would circulating RNA or RNA from white blood cells contaminate RNA in exosomes?
It depends on your process. If you, like I said, if you activate thrombocytes, that will definitely affect your result. RNA that leaks out from lymphocytes when you do the blood collection is typically degraded if it leaks out by lysis, because free RNA does not exist in plasma, or cannot exist in plasma. Free RNA is degraded within seconds. The only way an RNA would survive is if it's protected by some means, either in a vesicle or in a protein complex or a lipid complex.
Okay, the last question before we close. Does each individual have a unique exosome baseline?
Yes, there seem to be, so we have only limited data on that, but there seem to be um, a unique pattern in certain individuals. So when we did profiling of longitudinal samples from a cohort of patients, we could actually group the pre and the post sample from the same individual, so they looked similar to each other. So there's definitely a unique profile that can be found, yes.
Okay, thank you so much, Johan. So I need to cut the Q&A at this point to respect everyone's time. I want to thank all the participants to stay online, and again, thank you, Johan, for a very informative, enlightening presentation. And please look out for an email with a link for the presentation, as well as the written responses for the questions. And also remember that this is the second session of 4, so please don't forget to register for the upcoming sessions. And thank you very much.
Thank you.
Joanne?
Joanne, we're not hearing you. Maybe you're muted still.
I can still hear you.
Hello there, this is Joanne Gear, and I want to thank everyone for being here. The system's closed and opened again for me. This has been a really terrific program. We're looking forward to next week, and we will be sending out an email with follow-up for the recording and so on. Thank you, everyone.